Good afternoon, students. We are discussing about transcription in the last class. You said that transcription is a process of synthesis of mRNA. The mechanism of synthesis of mRNA from the DNA is declared as transcription. Okay, so transcription process. Transcription process occurs where? Okay, it occurs at a site which is called as transcription unit. So now where is transcription unit? So transcription unit means it was having three important locations. You said that the transcription unit has promoter site. So where is promoter site means 5' to 3' upstream end. The promoter site 5', 3' upstream end, terminator site 5', 3' downstream end. Yes, this chain is non-template. This chain is a non-template strand. It is also called as coding strand. This is template strand. Template strand. So what is promoter site means? The point where transcription starts. Terminator site, the point where transcription stops. Then where does transcription occurs? In this site which is called structural gene. Structural gene. So what is structural gene? We call structural gene as is called as a cistron. Structural gene is declared as a cistron. Means cistron is a structural gene or structural gene is called as cistron. Both are same words. Ultimately what it is? It's a part of the DNA who codes for a polypeptide, who makes an amino acid chain, right? So it's a part of the DNA, right? Gene only. Structural gene is a part of DNA. Structural gene is part of DNA itself, who codes for a polypeptide, who codes for a polypeptide, right? It's a part of the DNA who codes for a polypeptide itself. Structural gene is equal to cistron, a cistron who part of the DNA who codes for a polypeptide. Now we'll see in prokaryotes. In prokaryotes, the structural gene is one or it is in many. Promoter और टर्मिनेटर के बीच में जो भी स्ट्रक्चरल जीन है वो एक है या ज़्यादा है, वो ज़्यादा है. There are many genes make many mRNA, therefore you call it as polycistronic. So in prokaryotes, the cistron contains many genes. Cistron has many genes. Cistron has many genes. Many genes means many polypeptides. Many genes, many polypeptides. Means many different forms of proteins. Therefore, it is called as polycistronic. Polycistronic. In case of eukaryotes, the cistron, it has only one gene. The structural gene is only one. As it is on the board, promoter, terminator के बीच में there is only one gene. But in prokaryotes, there are many genes. Many genes, many mRNA, many polypeptide chain, many proteins. One gene makes one mRNA makes one polypeptide chain. So in eukaryotes, cystone has only one what? One gene. Cystone has only one gene. Therefore, cystone has only one gene. Therefore, it is monocystonic. It is monocystonic in a condition. Right? Cystone has only one gene. Monocystonic in eukaryotes. Polycystonic in prokaryotes. Polycystonic in prokaryotes. Monocystonic in eukaryotes. So what is cystone? Cystone is nothing but a structural gene only. What? The gene or a cystone is nothing but the part of the DNA who makes or who codes for a polypeptide. So now we will see what are the steps in the transcription. Transcription kaisa hota hai? Which are the 3 steps involved in transcription. So we'll complete the process of transcript. Three steps involved in a transcription: initiation, elongation. Third step is termination. Initiation, elongation as well as termination. So this could be the 3 steps in the process of transcription: initiate, elongate and terminate. So what did you mean by the word initiation? So how does initiation occurs? How does initiation occurs? Here, first enzyme RNA polymerase binds to this site which is called as promoter. Okay, with the help of the enzyme RNA polymerase, and because of the factor called as sigma factor. A enzyme के अंदर a factor is there which is called as sigma. Enzyme has a factor sigma. This makes the enzyme to attach to this site of transcription unit which is called as promoter. Which is called as promoter. So here number 1, initiation. Here, so what happens in the initiation means here the enzyme. What did you see on the board there? The enzyme, right? What? RNA polymerase. Here, the enzyme called RNA polymerase binds to the promoter site. Here the enzyme RNA polymerase binds to the promoter site with the help of sigma factor. With the help of the sigma factor, enzyme RNA polymerase binds to the promoter site with the help of a sigma factor, right? This is called as initiation. It just goes and binds. To which part of the DNA? To the template. To the template form of the DNA. Next, B, elongation. You will add the RNA nucleotides along the length of this DNA. You keep on adding the RNA nucleotides along the length of the DNA because transcription is a synthesis of mRNA from the DNA chain. Synthesis of mRNA from the DNA chain is called as transcription. Here, hoping a chain is a CGACTTAGCT. Okay, now you start adding nucleotides along the length of this DNA. You start adding the new nucleotides along the length of the DNA. C, G, G is C, A is U, C is G, T is A, T is A is U, because thymine is replaced by uracil, again C is G. So this is how make a long chain of mRNA. So this occurs in the region called structural gene. This process is called as elongation. जो भी न्यूक्लियोटाइड्स डीएनए टेम्पलेट के आगे हम जोड़ते जाते, राइट, उसको ऐड करते जाते, that will be declared as a process what you call elongation. So what will you write? Elongation को क्या लिखना हम? I'll write for elongation here. The core enzyme. The core enzyme, RNA polymerase. What do you mean by the word core enzyme? Means now without sigma factor, only the RNA polymerase enzyme. Right? Now it is for attaching you want sigma. Later sigma is not needed, so it just adds the DNA template ke aage an mRNA chain. So here the core enzyme RNA polymerase. Here the core enzyme RNA polymerase. What will you do? Means it adds the RNA nucleotides. Adds the RNA nucleotides across the structural gene. Across the structural gene, the core enzyme RNA polymerase adds the RNA nucleotides across the structural gene. So this is about what? The process called as elongation. Right? Core enzyme RNA polymerase adds the RNA nucleotides along the length of the structural gene. So this is number 2, elongation. Now last, third step, what is called as termination. Right? So now the mRNA synthesis has come to this site called as termination site. How will you stop here? Means by addition of the factor called as rho factor. Right? So in the third step, termination. In the third step called termination, jo bhi aap bole. So what you are supposed to write in termination means so in termination, the RNA polymerase gets bind to rho factor where this is at a terminator site, right? As you can see here, this is at which site means RNA polymerase enzyme will be attached, gets bind to this, which is called as a rho factor. RNA polymerase gets bind to this rho factor at which site? At the terminator site. At terminator site. Okay? To stop transcription. Why means to stop the process of transcription. To stop the process of transcription. Now very simple. This mRNA will live between the two DNA chains. Whatever mRNA will be released that comes into the cytoplasm in prokaryotes, because there is no nucleus. Replication, transcription, translation all occurs in the single cell only, because in eukaryotes you have nucleus. Replication and transcription occurs in the nucleus. Translation occurs in the cytoplasm. Translation occurs in the cytoplasm, but replication and transcription occurs in the nucleus. So now this is what we got: a primary transcript. Okay. Now, in prokaryotes, further, what is to be added in prokaryotes? Now, this is a primary transcript. This is primary transcript. So, we are talking in prokaryotes. In prokaryotes, we are talking about a primary transcript. So, in this primary transcript, should we cut off any part called as introns and join the exons? Means no, right? It's not like that. So, primary transcript, there is no splicing of, there is no splicing of introns and joining of exons. There is no splicing of introns and joining of exons. ऐसा नहीं होता that introns को हम तोड़ देंगे और exons को join, ऐसा cutting and pasting कुछ भी नहीं होता in prokaryotes. Whatever mRNA has come, the primary transcript, it remains as it is. It remains and functions as it is. Cutting and joining नहीं है. और दूसरा, इसमें कुछ capping, tailing ऐसा modifications भी नहीं है. And the primary transcription does not undergo it does not undergo any modifications. It does not undergo any modifications. It does not undergo any modifications. That is also there, right? Capping, tailing, and last thing, sir, वो जो mRNA बनाए, RNA polymerase enzyme, whatever RNA polymerase enzyme has made this mRNA, whether any other RNA polymerase is there to make tRNA or rRNA, means answer is no. All mRNA, tRNA, rRNA are made by only one kind of RNA polymerase. Last sentence. So, what shall I write here? So, we shall do right after the transcription. To be noticed, in prokaryotes, mRNA, tRNA, rRNA molecules, mRNA, tRNA and rRNA molecules, all, right? mRNA, tRNA and rRNA molecules, all are synthesized by, all are getting synthesized by whom? All are getting synthesized by only one type of or same type of RNA polymerases, right? All are going to be synthesized by same type of RNA polymerase. Only one kind of RNA polymerase it is. So, in case of eukaryotes, there are different types of RNA polymerases. Polymerase 1, polymerase 2, polymerase 3, so whatever. So, what has to be done now? Nothing. We will discuss about transcription in eukaryotes, right? How does transcription occurs in eukaryotes? We shall focus this in the next class. So, how does it occurs? How does it occurs? We shall see this in the next class. Now, we will make a small revision and we'll watch some animated videos for your coming test. I hope that will help a lot. So, rather than discussing MCQs, we'll show you some animations perfectly which is connected to your syllabus. So, I know you to have in your respective YouTube channel so many animated videos, but one which suits you, right, for very simple and short, very authenticated to deal with the replication process, transcription process. Okay. So, now here, what was the 3 steps of transcription? It is still on the board here. So, first, here enzyme RNA polymerase, the enzyme RNA polymerase, it binds to the promoter site, correct, with the help of the factor called as sigma factor. Then what happens? The 2 DNA chains will open, right? Without that's not possible. The 2 DNA chains will open by breaking of hydrogen bonds. This is the template strand. This is non-template. This is called as non-coding strand. This is called as coding strand. Sir, why did you call this as coding strand? Because this mRNA chain and these DNA both have same set of nucleotides. जो भी mRNA है, the same sequence of nucleotides is also found in this part of DNA, right? Because two DNA are complementary. So पहले के सामने और एक नया mRNA बना है तो भी जो भी mRNA बनाए और जो भी नीचे ये DNA पार्ट है दोनों में न्यूक्लियोटाइड सीक्वेंस एक ही है, सेम ही है. इसलिए जब mRNA is sense, it takes information. So same sequence is found on this also. So you have to call this as sense strand. नीचे वाला sense कहलाता है. mRNA codes for amino acid. नीचे वाला भी having the same set of amino acid sequence. So therefore it is also called as coding strand, right? mRNA language और नीचे वाला DNA language दोनों भी same है. Coding, इसको भी coding बोलते हैं, sense और इसको भी sense बोलते हैं. This is the reason why this lower chain of DNA is called as coding strand. Okay, that's all the reason is. But template is only one. ये ऊपर वाला जो भी है वो template है. He will make this part of mRNA complementary role. हमने कुछ यहाँ पे दिखाया आपको. C, G, G, C, A, U, C, G, T, A, T, A. Okay, so this is like this. Over. So second step. What was second step? It is elongation. So what is elongation? Here the core enzyme RNA polymerase. The core enzyme RNA polymerase will add the RNA nucleotides across the structural gene. This core enzyme means without sigma factor, enzyme starts adding the nucleotides along the structural gene. So the second process is called elongation. Third, termination में क्या होगा? To this RNA polymerase enzyme, you will bind a factor whose name is called as a rho factor. You will bind a factor called as rho factor. जब rho factor will bind, so transcription will stop, right? So that's the last part of transcription unit. Transcription will stop. So the RNA polymerase becomes free. RNA polymerase becomes free, and mRNA is called as primary transcript. इसमें in prokaryotes primary transcript में exons, introns ऐसा कुछ भी नहीं होता. Introns are removed, exons never happens, and यहाँ पे capping हुआ, tailing हुआ ऐसा कुछ भी नहीं है, because it is a polycistronic. Many mRNAs will come here. I have shown only one on the board. Many such mRNAs will come here as shown in the same procedure. Okay, so no changes in that mRNA. So all transcription, translation, replication, everything occurs in the cytoplasm. So this is the transcriptional differences in prokaryotes and eukaryotes. Abhi eukaryotes, transcription in eukaryotes, we shall see this in the next week again, so that you will have a revision of this. So now I will stop here, so you can watch the videos which will be telecasted right now. So after that, anyhow, next week again we will be back.
Themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, 2 identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
Hey everyone, quick biochemistry basics here. Let's talk about Messelson and Charles' experiment. Watson and Crick had a hypothesis that the DNA replication was semi-conservative, However, there was no experimental evidence to test this hypothesis. So, to test this hypothesis, Messelson and Stahl in the year 1958 designed a wonderful experiment. Now, the word semi-conservative means each of the strands is going to act as a template for the synthesis of new strand. And to prove if this phenomenon is true or not, we must have a method to distinguish between the parent strand and the daughter strand. Yes, distinguish between parent strand and daughter strand. So here is what Messelson and Stahl did. They took E. coli wild-type cells and grew them in media containing N15 for several generations. N15 is a non-radioactive heavy isotope of nitrogen. The presence of N15 will make all the molecules in the cells, including the DNA, heavy. What does it mean by heavy DNA? It simply means if I take E. coli cells grown in N15 media and subject its DNA to density gradient centrifugation, then the density of this DNA will be more than the density of the DNA taken from cells grown on N14 media. The cells grown in N15 media were collected and transferred in fresh N14 media. So as the cells divide, they incorporate N14, which is the normal isotope, into its DNA. The cells were harvested every 20 minutes and the DNA was isolated and subjected to density gradient centrifugation. Let's take a look at the results. The presence of only intermediate band after the first generation indicates that the heavy strand was acting as a template for the synthesis of new strand with N14 isotope. As the cells divide further, more and more N14 is incorporated into its DNA. These results indicate that the replication of DNA is semi-conservative.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. The motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then, during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins